Alright guys, so today we're gonna talk about the mitotic phase of the cell cycle and go into detail about the process called mitosis. Here we go. And so mitosis is the cell reproduction for any cells in your body that aren't sex cells. And it's pretty simple really, it's only 4 steps. Or 4 stages, we like to call it. And they're called prophase, metaphase, anaphase, and telophase. These are the stages that pull our chromosomes apart into 2 sets, followed by the second part of the mitotic phase, called cytokinesis, which separates the cytoplasm, and thusly creating 2 new cells. But we'll get into that later. Okay, so we're gonna start it off by talking about the first part. And this one is called prophase, like I mentioned. Prophase. And prophase is really when the chromatin in the nucleus condense, and they're actually visible under a microscope. They become so large as chromosomes. So I'm going to draw a few right now. Here's one. And I'll draw a blue one over here. And another one, I'll choose green. Okay. So those are our 3 chromosomes. And notice how there's no nucleus here. I could draw a kind of outline, but really at this stage, the nuclear membrane, it dissolves at this point so that we can undergo the stages of mitosis. And the last thing in prophase is that we're going to include the things we like to call the microtubule organizing centers. So I'm just going to draw one there and there, on opposite sides of the cell. And what they're going to do later on is, well, you're going to see it in the next stage called metaphase, which we're moving on to. See, that's the arrow, and meta. Oops. Okay, sorry about that. But it's a metaphase. And a good way to think about this is, notice the M. Think of it as the middle, because, watch for a second. We're going to look at the microtubule organizing centers again, over here. And they're actually going to attach to the chromosomes. And by doing that, they're also going to push them towards the middle of the cell. So the chromosomes will be all lined up in the middle. Oops, that one's actually blue. And the green. And all of them are lined up and attached to the spindle fibers that come out of the microtubule organizing centers. Those are just the strings that attach from them. And that's pretty much all that really happens in that stage. So now we're going to move on to the next one. Oops. That we like to call, or that it rather is called, anaphase. And in this phase, really, it's just the spindle fibers are beginning to pull apart, or rather they do pull apart the chromosomes in half, which each spindle fiber will take the corresponding part of the chromosome, or the chromosome. and they're pulling it towards their sides of the cell at opposite sides. And now we're going to move on to our final stage, which is called telophase. And in this stage, the spindle fibers are really removed, and the chromosomes are just sitting comfy on their sides of the cell. There we go, and the green. And at this point, the nuclear membrane will reform, but it'll reform around each area where there was a microtubule organizing center. So now there's going to be 2 nuclei in this cell. But wait, now we have 2 fully functionally able to become 2 cells. Or wait, that came out wrong. It's what we need for 2 functioning cells that we have. But how do we separate this into 2 fully functioning cells? Well, that's for our last step, called cytokinesis. And cytokinesis is different depending on what cells you're looking at. Like for the cell that we've been looking at, it's a round cell, which means it doesn't have a cell wall like plant cells do. So we've been looking at an animal cell this time, right? But we'll talk about the animal cell first for cytokinesis, okay? So I'm going to show you that it's going to start to pinch off in the middle. With our 2, they're fully formed nuclear membranes. And they have the chromosomes in it. It's fine. And the one thing that's pinching it off in the middle is called a cleavage furrow. And it forms by microfilaments. And this pinches tighter and tighter until we get our 2 new cells. Let me just... And they have their nuclei. And they're fully functional and ready to go, right? But for plant cells, it's a bit different. Let me just get a plant cell. Look at those sharp edges. Okay, so here's a nucleus here. Nucleus there. There, yeah. And alright, we're already starting to see some cell platelets form in the middle. This will be the thing that separates the 2 cells from each other, and now they're 2 fully functioning cells as well. Look at that. And that's all there really is to it, so thanks guys for sticking with me.